Assalamualaikum, I'm Insyirah binti Ismail, and today I am going to present my project entitled Energy Efficient Harvesting of Spirulina using a tilted panel membrane filtration. So before I proceed any further with my presentation, I would like to give an overview of today's presentation. So firstly, I will talk about the introduction and the literature review, followed by the methodology, and next I will elaborate on the results, and lastly I will talk about the conclusion. So for the introduction, the chemical or the synthetic colorant have been restricted by almost every authorities all over the world. This is due to the fact that the chemical colorants impose bad health effects to the humans and also could trigger the allergic reactions to the humans. Therefore, a natural resource such as spirulina is an alternative to this issue. So spirulina is actually a superfood because it contains excellent nutritional contents such as vitamins, minerals, proteins, polysaccharides, and many more. And because of its excellent nutritional contents, it is suitable to be processed into food and beverage production and also pharmaceutical products. So for example, for the first one is the pharmaceutical products, the polysaccharides from the spirulina can be extracted and formed into tablets and pills in order to reduce the growth of cancerous cells and also to prevent the HIV infection. Besides that, the amino acids from the spirulina can also be incorporated into sports drinks in order to improve the performance of the athletes. And other than that, the spirulina can also be incorporated in the commercial food production, such as in one of the Malaysia crackers company, which is the Lee Biscuits company, actually uses the spirulina in their crackers production in order to improve the digestibility of the crackers. And besides that, the M&M company also uses the natural colorant from the spirulina in their chocolate production. So there are actually several options of spirulina harvesting methods such as sedimentation, coagulation, centrifugation, and also filtration. And among all these harvesting methods, the filtration methods is the most suitable option to harvest the spirulina because it is a low cost option and it doesn't require any chemical addition in the process. Besides, since spirulina is involved in the production of high value products, so it demands almost no chemical contamination in the process. Therefore, filtration is the most suitable for the spirulina harvesting process. However, one of the problem is the membrane fouling. So basically, membrane fouling occurs when the membrane surface is covered with foulants, which could reduce the permeability over time. And the membrane fouling can be categorized into its recoverability. So the first one is the reversible fouling. The example is the cake layer formation, and it can be removed by physical cleaning. As for the second one is the irreversible fouling, and it can be removed by chemical cleaning. And the examples are pore blocking and also surface fouling. And the last one is the irrecoverable fouling, which could not be removed neither chemically nor physically. So one of the ways in controlling the membrane fouling is to implement the air bubbling system. So basically the air bubbles will be supplied at the bottom of the tank, and the air bubbles will scour off the foulant that form on the membrane by using the hydrodynamic force such as drag force and also impact force. So there are actually several configurations of the air bubbling system, which are vertical membrane panel, horizontal, and so tilted membrane panel. So for the vertical membrane panel, it is claimed that it has low efficiency because the bubble tends to move the furthest distance away from the membrane panel, causing low contact between bubbles and the surface of the membrane. As for the second configuration, which is the horizontal membrane panel, it is claimed that the bubbles and the membrane surface, the contact is maximized. However, it requires large footprint. As for the tilted membrane panel, it is claimed to have good contact between the air bubbles and also the membrane surface at a certain angle, at a certain tilting angle. And then move on to the summary of the literature review. So this table actually comprises of several fouling management systems which improve air bubbling system. The next one is the dynamic filtration system, and the third one is the physical cleaning system. So the main key points that we can extract from that table of literature review are that the first one is the most of the previous studies was conducted by using the same type of microalgae species such as Euglena and Chlorella. And the next one is the most of the studies were usually conducted by using one type of membrane, which was PVDF, and most of the studies previously was conducted at one value of concentration only at one time, which either could be 0.6, 0.65 or 1 gram per liter, and only a few studies investigated the energy analysis and no previous reports on the aeration energy analysis. As for the problem statement, this problem actually arises when the cake layer actually contributes up to 80% of filtration resistance, which could affect the permeability of the membrane. And there was actually previous reports on the tilting panel system, and the previous report was only conducted from the tilting angle from 0 up until 20 degree only, so there was no studies conducted beyond the tilting angle of 20 degree. And besides that, the most of the studies was conducted only by using one type of membrane, and there were only few studies analyzed the energy consumption of the system. And that problem statement leads to the objectives of this project. The first one is to fabricate and characterize of both membrane, and the second one is to compare the performance of the both membrane, and the third one is to evaluate the effect of aeration rate in the PVDF, and also to study the effect of microalgae concentration in the PVDF. And also the last one is to analyze the energy consumption of the tilted panel system and compare it with other literature. So these are the scope of study. So the parameters for this study is actually using spirulina, and also there are two types of membrane, and there are several tilting angles and aeration rate and also feed concentration that were used for this project. As for the methodology, this is the project workflow. So firstly, the membrane was prepared and characterized. So basically the PVDF was characterized, either it could be MF, microfiltration, or ultrafiltration. And after that, the filtration setup was prepared. So basically the spirulina broth was actually cultivated in a lab scale production. And after the filtration setup was prepared, the filtration test was first conducted by comparing the performance between PVDF and PSF membrane at different tilting angles, and we identify which membrane has better permeability, and that better membrane, which was PVDF, was continue to study the effect of other parameters such as aeration rate and also microalgae feed concentration. And all the data collected was analyzed by using the energy estimation and also the statistical analysis. As for the experimental setup, this is the setup of the experiment. So this tank is actually filled with 7 liter of microalgae spirulina broth. So there are actually two pumps here. So the vacuum pump and the air pump will be turned on simultaneously for 9 minutes. And after 9 minutes, the vacuum pump will be turned off for 1 minute in order to collect the permeate and in order to measure the permeate collected during that 9 minutes. And after 1 minute has ended, the permeate will be returned back into the feed tank, and the 9 minute of filtration will be resumed, meaning that the vacuum pump will be turned on. So this cycle of 9 minutes on and 1 minute off was repeated until 1 hour of experiment. And the performance of the membrane was actually evaluated by calculating the flux and the permeability of the membrane. So these are the equations that we use, and based on the data collected, we analyze the energy consumption by using two approaches. We analyze in terms of the total energy consumption by using this formula, and we analyze the aeration energy consumption by using this formula. As for the result and the discussion, there are two types of membranes that were used in this study. So for the first one is the PVDF membrane. So PVDF and PSF have different pore size, and PVDF has larger pore size, which makes it a microfiltration type, and PSF is a ultrafiltration type. And since PVDF is under the category of microfiltration type, it has larger pore size, which means that it has lower intrinsic mass transfer resistance than the PSF membrane. And which explains why PVDF has higher clean water permeability than the PSF membrane. Besides that, we can observe that spirulina cell size is 40 micrometer, which is actually larger than the pore size of the membrane, and meaning that the spirulina can be fully retained by both membranes. And apart from retaining the spirulina cells, it is also attractive to recover EPS. And according to a literature, the MF and UF membrane was actually can actually be combined to recover the EPS from the microalgae culture medium. Next, I will explain the comparison of PVDF and PSF membrane. So, as shown in this figure, the grey bar graph represents the steady state permeability for PVDF against tilting angle, and for the yellow bar graph represents the PSF membrane against the tilting angle. So from both figures, we can observe that both of the membranes have similar trend of permeability performance, because as the tilting angle increases from 0 up until 45 degree, the permeability increases. However, it starts to drop as it reaches 70 degree. And it is the same trend for PSF as well. So we can deduce that 45 at the tilting angle of 45 degree has the highest permeability, followed by 70, 20, and lastly 0 degree. However, this pattern actually contradicts with the sinusoidal pattern, because according to the sinusoidal pattern of impact force, at the tilting angle of 90 degree, the impact force is at the maximum value, which means that the permeability is also expected to be at its maximum value as well. However, the results showed otherwise, because the permeability starts to drop as it reaches 70 degree. And the permeability at 70 degree here is lower than the permeability at 45 degree, and it could be due to the overcrowding of the bubbles that could disrupt the permeate flow, hence lead to the permeability decline for the both of the membranes. And when we take a closer look at the maximum permeability for the PVDF and the PSF membrane, we can observe that the PVDF membrane, the maximum for permeability of PVDF is around 300 something, and the PSF membrane is around 100. So there is a huge difference between these two maximum permeability, and there is actually 62% of permeability difference between these two membranes. And it actually could be due to the larger pore size of the PVDF membrane, because when it has larger pore size, it has higher fouling propensity. And this also actually implies that this system is good for membrane with higher fouling propensity, because even though it has higher fouling propensity, it can still yield a high value amount of permeability. Therefore, the PVDF at the tilting angle of 45 degree was selected for investigating the next parameter. As for the effect of aeration rate, we can observe that as the aeration rate increases from 0.5 to 1 liter per minute, the permeability also increases. However, it reached its threshold value at 1 liter per minute. This is due to the fact that when we increase the aeration rate beyond 1 liter per minute, there will be no significant increase in the permeability. So there was no any need to increase the aeration rate beyond 1 liter per minute. And if we increase the aeration rate beyond 1 liter per minute, it could actually trigger the spirulina cells to release algae organic matter, AOM, which actually could make the membrane fouling to become much more worse. So therefore, the optimal aeration rate for this system is in the range of 0.5 to 1 liter per minute. And when we take a look at the permeability from 1 to 1.5 liter per minute, we can observe that there is a threshold permeability which is around 500, and this threshold permeability is actually about 68% of the clean water permeability. And this actually indicates that the foulant that was removed by the aeration supplied was actually the reversible fouling, and the remaining one, which was the irreversible fouling and the irrecoverable fouling, was not recovered by this aeration supply. So the irreversible fouling could actually be removed by conducting chemical cleaning in order to restore the membrane permeability. However, the irrecoverable fouling could not be restored neither chemically or nor physically methods. Next, for the effect of feed concentration, at the microalgae concentration of 2.15 gram per liter, we can observe that it has the highest permeability, and at 2.554 gram per liter, it has the lowest permeability. So we can deduce that as the microalgae concentration increases, the permeability increases. And at high feed concentration, it actually implies that accumulation of EPS is occurred, because EPS is actually one of the components in the spirulina, and it could actually impede the flow of permeate from passing through the membrane. Therefore, it is vital to identify the optimum concentration in order for the system to operate efficiently. Next, for the energy consumption and compare it with other literature. So, in terms of energy, we can observe that this study has the lowest total energy consumption, which is 0.2 kilowatt hour per cubic meter, and it has the lowest aeration energy consumption, which was 0.03 kilowatt hour per cubic meter. And if this value is used to calculate the full scale of energy consumption, the total energy input would be further reduced, because the aeration energy actually makes up the majority of the total energy consumption. Next, in terms of the permeability, and in order to compare with other studies, we can observe that when we compare this study with other types of membrane fouling control systems, such as dynamic membrane filtration and also physical cleaning, we can observe that this study is actually 20.4% higher and also 10.8% higher. So, when we compare with the fin spacer filtration, which is actually a type of air bubbling system as well, it is actually 36% lower than the fin spacer filtration. And this fin spacer filtration actually filter the chlorella microalgae species. Therefore, this is not a direct comparison because of its difference in microalgae species, operational parameters, and configuration. However, this study can be considered within the upper range of the results reported previously. Next, for the conclusion, we can observe that these are the objectives and the results for each of these objectives. So for the first objective is to characterize the PVDF and PSF, and we managed to do that because PVDF is the microfiltration and the PSF is the ultrafiltration, and it is proven that PVDF has higher clean water permeability than PSF because of the difference in pore size. And the next objective is to compare the performance of PVDF and PSF membrane in the tilted panel system, and the results obtain that permeability increases from 0 until 45 degree, and it starts to decline at the tilting angle of 70 degree. And when we compare between those two membranes, the PVDF has better membrane performance in terms of permeability by 62%. And the next objective is to evaluate the effect of aeration rate, and the results obtained was that the optimal aeration rate for this system is in between 0.5 to 1 liter per minute. And the fourth objective, which was to study the effect of microalgae concentration in the tilted panel system by using PVDF, we can observe that the highest microalgae feed concentration shows the least permeability, and the feed concentration in this case should allow the filtration to operate effectively. So we need to identify the suitable concentration for this system. And the last objective is to analyze the energy consumption for this system. So basically, this study has low total energy consumption and the lowest aeration energy compared with other studies. So these are the list of reference that I did for this project. With that, I end my presentation. Thank you.